Hello friends, welcome to BMH Learning. This video is about Luciferase assay. First of all, let's discuss about what is Luciferase. Luciferases are a class of oxidative enzymes that occur in several species. The enzyme enables the organisms that possess it to bioluminescence, or we can say it makes them to emit light. One of the most famous of these enzymes is the firefly luciferase. Fireflies have the ability to emit light through a chemical reaction in which luciferin is converted to oxyluciferin with the help of luciferase enzyme. Now the reaction catalyzed by the luciferase is highly energetically efficient. That means all the energy supplied into the reaction is rapidly converted to light. Because of this, the enzyme is extremely sensitive, which makes it a great candidate for a reporter assay. Now let's know what is a luciferase reporter assay. A luciferase reporter assay is a kind of test which determines whether a protein can activate or repress the expression of the gene of interest with the help of luciferase that is used as a reporter protein. Now, in order to perform the luciferase reporter assay, first thing we need is the gene of interest. Then we have a suitable expression vector here. The significance of this vector is that it has a regulatory element or a promoter region. This region is fused with a DNA sequence that actually codes for a luciferase enzyme. So once activated, the system would produce luciferase. Now, look back at the gene of interest. The gene has a regulatory region which would turn on or off the transcription. So what we do is, we clone the regulatory sequence of gene of interest upstream of the luciferase gene fused in the expression vector. To make it simple and easy to understand, I am not showing entire vector, only important components are mentioned. In the next step, we transfect this recombinant construct into a host cell to increase copy number. It's allowed to grow for a few days on suitable nutrient media. Once the promoter on the vector is activated, this would turn on the transcription of the luciferase reporter gene, and we get the luciferase protein. In the next step, cells are lysed using an appropriate cell lysis buffer. So we get cell lysate. Cell lysis break the cells and release all the proteins including the luciferase. The cell lysate is transferred to a multi-well plate. Into the cell lysate, we add the substrate, that is luciferin, and all the necessary cofactors. The luciferase will catalyze the reaction and the light is emitted. The light is measured using a luminometer. Luciferase activity is based on the amount of bioluminescent signal. Now, the importance of this assay is that it establishes a functional connection between the presence of the protein and the amount of gene product that is produced. It's because the target gene is fused to the luciferase reporter gene. Thus, the luciferase activity can be directly correlated with the activity of gene of interest. This was all. Thanks for watching.